would like to present the results of the research regarding synthesis of a novel porous materials with hydrophilic properties with the use of ethyl methacrylate and glycidyl methacrylate components. The main goal of our project was the synthesis of the oligomeric network of polyethyl methacrylate co-oligomer glycidyl methacrylate, for which we used the well-known oligomers containing ethyl methacrylate and glycidyl methacrylate components. In order to change the hydrophobic character of hema co-oligomer to a more hydrophilic one, the oligomeric chain was functionalized with ethanolamine, xylitol, and ornithine. The first step in hydrogels preparation was the synthesis of co-oligomer and their covalent functionalization with hydrophilic moieties. To obtain the desired hydrogels, a suitable oligomer consisting of two components, ethyl methacrylate and glycidyl methacrylate, were prepared. Then the oligomeric chain was modified with ethanolamine, xylitol, and ornithine to obtain the hydrophilic 3D co-oligomers. The standard method for the preparation of methacrylic acids, ester oligomers or polymers is based on a radical polymerization reaction initiated with ABN, usually carried out in the toluene solution, followed by precipitation of the resulting oligomers or polymer with methanol and further purification of the product. Herein, the reaction was performed in heptane solution that reduces the dispersion of the obtained product due to the dependence between the length of the oligomeric chain and its solubility in the reaction medium. The longer chain of the oligomer reduced the solubility of the oligomer in the reaction medium. This process made it possible to obtain an oligomer using precipitation polymerization. The experimentally selected molar ratio of ethyl methacrylate and glycidyl methacrylate and initiator ABN was 1 to 2.6 to 0.2, respectively. Further, the functionalization of co-oligomer was performed using a method of post-modification of co-oligomer with application of microwave-assisted synthesis. The procedure was based on the covalent reaction of co-oligomer and reactant ethanolamine, xylitol, or ornithine, dissolved in a different medium. Research on the synthesis and modification of PGMA chains through reaction with various azanucleophiles allowed to state that they proceed mainly in the direction of epoxide ring opening. Fourier transform infrared spectroscopic method was used to analyze the chemical structure of the obtained oligomers. Here we can see the spectra of the non-modified and modified oligomers of PMA's co-oligomer GMA for the non-modified oligomeric networks. Two strong characteristic peaks were observed at about 1725 and 1150 wavelength. The oligomers show the bands corresponding to the stretching vibrations at 910 and 850 wavelengths. The epoxy group in the polymeric network gave an absorption band due to the vibration near 915 wavelength. The extent of epoxy can be monitored by measuring the intensity of this band. In order to confirm the proposed synthesis pathways of the oligomers, the molar mass distribution and the average molar mass of the methacrylate oligomer and the derivatives were determined using the size exclusion chromatography technique. The results of determination of the average molar mass for the methacrylate oligomer derivatives obtained with the use of size exclusion chromatography are visible here on in a table 1, while the molar mass distribution of the fraction tested will be presented in our publication. The molar mass distribution for the obtained oligomers and the dispersity index with a value close to 1.1 indicate that the oligomers which were synthesized are homogeneous material with a fairly homogeneous distribution of the length of oligomer chain. Average molar mass values for the methacrylate oligomer derivatives calculated based on size exclusion HPLC method clearly indicate that the resulting chains are short and have about 3 or 4 mers in the oligomer. Based on the results obtained by size exclusion chromatography and 1H NMR spectra, the main conclusions can be made as follows. First, for co-oligomer, the number of EMA to GMA units is about 1 to 2. Second, the co-oligomer is incompletely functionalized with ethanolamine, the presence of two singlets in NMR spectrum proved the incomplete conversion of the epoxy groups into functionalized co-oligomer. The number of EMA to GMA units in co-oligomer is approximately 1 to 2.5. Next, the reaction of co-oligomer with xylitol under the influence of DBU used as a catalyst gives a compound with a mass of 713. The functionalization reaction takes place with the opening of the epoxide group as well as the transesterification of the glycidyl group. Differential thermogravimetric measurements were performed to analyze the thermal stability of the non-modified and the modified co-oligomer. Almost all post-modified oligomers show higher thermal stability than the starting materials, which confirms the modification of the epoxy groups with ethanolamine, xylitol, and ornithine, respectively. These results clearly show that the thermal stability of the obtained oligomers is related to the chemical structures of the chains and the method used for the functionalization with hydrophilic moieties. Synthesis of the co-oligomer and the further modification with moieties containing hydroxyl and amino groups allow to obtain the products in the form of white or slightly yellow solid powders. The key to the successful preparation of the modified co-oligomer is a judicious selection of monomers and hydrophilic component. The resulting hydrogels are stable over time, but upon removal of the solvent from their networks, they reform a solid powder. Here are shown some images of films of the non-modified and the modified co-oligomer made by using scanning electron microscope. The morphology of the non-modified co-oligomer differs from the oligomers modified with ethanolamine, xylitol, and ornithine. The modification of co-oligomer with hydrophilic moieties increased their dispersibility in protic solvents, which is shown on figures 3C, D, and F. Only the functionalization of co-oligomer with ethanolamine leads to a reduction of the porosity of the obtained oligomer films, and the surface morphology appears more homogeneous. A characteristic feature of hydrogels is their ability to water uptake, which is called as a swelling behavior. As can be seen from the results collected in Table 2, the compounds marked in table as fourth has the best swelling properties. The test tube inversion showed that the compound first, second, and fourth form gels very well. Compound marked here as a second, which did not sufficiently gel to remain in the tube, nevertheless forms a dense structure that can be considered as a gel. For our applications where the absorption of water, which means swelling behavior, is not the most crucial, each of the oligomers can be used for further research. Due to the fact that the hydrogels used as a matrix for immobilizing of the active substances should have a compact structure of the polymer chain, where the polymer material must have adequate mechanical stability and stiffness, a small amount of 10% agarose was added for release of model compounds from hydrogels matrix. The study was carried out at 37 Celsius degrees, simulating the average body temperature. Methylene blue was used as a model compound to study the release from the gel matrix due to its hydrophilic hydrophilic properties and the easy determination by UV-vis spectrophotometric methods. The results showed that the release of methylene blue loaded into the hydrogel is highest and fastest during the first 6 hours. After this time, the release is reduced until it reaches the equilibrium with the solution, which was observed during 48 hours of the experiment. The obtained results indicate that the obtained oligomers can be used in the future to develop materials for controlled prolonged release of hydrophilic active substances. The cytotoxicity tests showed non-toxicity of only 2 non-modified compounds. Other samples were characterized by high cytotoxicity since cell viability was significantly reduced. In conclusion, we have demonstrated the synthesis of the oligomeric network, polyethyl methacrylate co-oligomer glycidyl methacrylate, which was performed to be used as a matrix for further functionalization. To change the hydrophobic character of the obtained co-oligomer to a more hydrophilic one, the oligomeric chain was functionalized with ethanolamine, xylitol, and ornithine. Synthesis of co-oligomer and their further modification with moieties containing hydroxyl and amino groups allowed to obtain the products in the form of white or slightly yellow solid powders. The modified oligomers in the form of hydrogels were used for the immobilization of methylene blue that was used as a model compound to study its release. The results show that the release of methylene blue loaded into hydrogels is highest in the first 6 hours. Based on a cell culture experiment, it may be concluded that only 2 obtained compounds have the potential to be used in biomedical application as drug carriers. The results indicate that the obtained oligomers can be used in the future to develop materials for controlled prolonged release of hydrophilic active substances. To conduct these studies, it would be not possible without the financial support of National Science Center and a group of people that work on this project. Thank you very much for your attention.